Fatty acid metabolism can feel like a maze of enzymes, cycles, and confusing regulation. But on the MCAT, it's actually one of the most testable, high-yield topics. What really happens when your body breaks down fat for energy, and how does it decide when to build it instead? In this video, we'll simplify fatty acid oxidation and synthesis step by step, connect them to energy yield, and highlight the key regulation points you need to recognize fast on test day. Fatty acid oxidation, also called beta oxidation, breaks down fatty acids into acetyl-CoA units in the mitochondrial matrix to generate energy. This process is central to how the body extracts energy from fats, especially during fasting or prolonged exercise. It begins with activation of the fatty acid in the cytosol, where fatty acids are converted into fatty acyl-CoA using ATP, forming AMP and pyrophosphate. This step effectively costs 2 ATP equivalents. Because long-chain fatty acyl-CoA cannot cross the mitochondrial membrane directly, the carnitine shuttle is required. The fatty acyl group is transferred to carnitine via carnitine acyl transferase 1, transported across the inner mitochondrial membrane, and then transferred back to CoA by carnitine acyl transferase 2. Once inside the mitochondrial matrix, beta oxidation proceeds through 4 repeating steps: oxidation, hydration, oxidation again, and thiolysis. Each cycle shortens the fatty acid by 2 carbons and produces 1 acetyl-CoA, 1 NADH, and 1 FADH2. These reduced coenzymes feed into the electron transport chain, making beta oxidation highly energy efficient. A common MCAT focus is calculating ATP yield from fatty acids like palmitate, which yields a large number of ATP due to repeated cycles. Remember that acetyl-CoA can enter the citric acid cycle or be used for ketone body formation during starvation, linking beta oxidation to other metabolic pathways. The carnitine shuttle is essential for transporting long-chain fatty acids into the mitochondrial matrix for beta oxidation. Without this system, fatty acid oxidation would not occur efficiently because the inner mitochondrial membrane is impermeable to fatty acyl-CoA molecules. The shuttle involves 3 main components: carnitine acyl transferase 1, a translocase, and carnitine acyl transferase 2. First, carnitine acyl transferase 1, located on the outer mitochondrial membrane, transfers the fatty acyl group from CoA to carnitine, forming fatty acyl carnitine. This molecule is then transported across the inner membrane by a translocase protein. Inside the matrix, carnitine acyl transferase 2 transfers the fatty acyl group back to CoA, regenerating fatty acyl-CoA for beta oxidation and releasing carnitine to be recycled. A key regulatory point is that carnitine acyl transferase I is inhibited by malonyl-CoA, a molecule involved in fatty acid synthesis. This prevents simultaneous synthesis and breakdown of fatty acids, which would be energetically wasteful. On the MCAT, you may be asked about this regulation, or about the consequences of carnitine deficiency, which can impair fatty acid oxidation and lead to muscle weakness or hypoglycemia. This shuttle also connects to broader metabolic themes like compartmentalization and regulation of opposing pathways. Each cycle of beta oxidation produces 1 acetyl-CoA, 1 NADH, and 1 FADH2, contributing significantly to ATP production. This is why fats are such an energy-dense fuel source compared to carbohydrates. The process consists of 4 steps. First, oxidation by acyl-CoA dehydrogenase produces FADH2. Second, hydration adds water across the double bond. Third, another oxidation produces NADH. And fourth, thiolysis cleaves the bond, releasing acetyl-CoA. Each of these products feeds into downstream pathways. NADH and FADH2 donate electrons to the electron transport chain. generating ATP through oxidative phosphorylation, while acetyl-CoA enters the citric acid cycle. A high-yield MCAT concept is calculating total ATP yield. For example, palmitate, a 1,6-carbon fatty acid, undergoes 7 cycles of beta oxidation, producing 8 acetyl-CoA, 7 NADH, and 7 FADH2. After accounting for activation energy, this yields over 100 ATP, far more than glucose. This highlights why fats are used for long-term energy storage. The MCAT often tests your ability to track carbon units and energy carriers, so practice counting cycles and products. Also, note the connection to fasting metabolism, where beta oxidation increases and supports gluconeogenesis by providing ATP and acetyl-CoA. Fatty acid synthesis occurs in the cytosol and builds fatty acids from acetyl-CoA using NADPH as a reducing agent. This pathway is essentially the reverse of beta oxidation, but takes place in a different cellular compartment and uses different enzymes. It begins with the transport of acetyl-CoA from the mitochondria to the cytosol via the citrate shuttle. Once in the cytosol, acetyl-CoA is converted to malonyl-CoA by acetyl-CoA carboxylase, a key regulatory enzyme. This step requires biotin and ATP and is the committed step of fatty acid synthesis. The fatty acid synthase complex then sequentially adds 2 carbon units from malonyl-CoA to a growing chain, producing palmitate as the final product. Each elongation cycle involves condensation, reduction, dehydration, and another reduction, using NADPH as the electron donor. NADPH is typically supplied by the pentose phosphate pathway, linking fatty acid synthesis to carbohydrate metabolism. On the MCAT, remember that synthesis happens in the cytosol, uses NADPH, and builds up rather than breaks down. A useful mnemonic is that synthesis is cytosolic construction, while oxidation is mitochondrial breakdown. Understanding these contrasts helps avoid common test traps. Fatty acid metabolism is tightly regulated to prevent simultaneous synthesis and degradation, primarily through malonyl-CoA and hormonal control. The body avoids wasting energy by ensuring that fatty acid synthesis and beta oxidation do not occur at the same time. The key regulatory molecule is malonyl-CoA, which inhibits carnitine acyltransferase the first, thereby blocking fatty acid entry into the mitochondria for oxidation. When malonyl-CoA levels are high, fatty acid synthesis is active and oxidation is suppressed. Hormones also play a major role. Insulin, released in the fed state, promotes fatty acid synthesis by activating acetyl-CoA carboxylase and increasing malonyl-CoA levels. In contrast, glucagon and epinephrine, released during fasting or stress, inhibit acetyl-CoA carboxylase and stimulate beta oxidation. These hormones signal the body to mobilize stored energy. On the MCAT, questions often test whether you can predict which pathway is active under certain physiological conditions, such as fed versus fasting states. This regulation also connects to broader metabolic integration, including gluconeogenesis, ketogenesis, and glycogen metabolism. Keeping track of hormonal signals and key intermediates like malonyl-CoA is crucial for mastering metabolism questions.